Hi, in this video, I'm going to talk about classical Mendelian genetics, or the genetics experiments that Gregor Mendel did with peas. After this lecture, you should be able to analyze the results of a genetic cross, and you should be able to do this to determine which allele or version of a gene is dominant over another allele, to predict the genotypes of the offspring if we tell you about the parents, predict the ratio of phenotypes in the offspring when we tell you about the parents, and potentially work backwards to figure out the parental genotype if we tell you about the phenotypes and the ratio of phenotypes that you observe in the offspring. And we've already briefly discussed that just the concept of genes, that they are the heritable unit of nucleotide sequence that encodes a functional product. So we've talked about how DNA genes can be transcribed to messenger RNA and translated to protein product, or some DNA genes are transcribed directly to a functional RNAs. Regardless, you have this DNA code that results in a functional product that affects the phenotype of that individual.
Before Gregor Mendel came along, there were a few theories floating about how genetic information was inherited. People could see with their eyes that offspring tended to look more like their parents than some stranger on the street, for example. So they knew that somehow information from parents was being conveyed to offspring, but they didn't know the mechanism. So here are a few hypotheses, some examples that people use to explain it. The first is preformationism, seen over here on the left, which believed that there were preformed little organisms. In the case of human, they called them homunculi, that exist inside gametes. This is a fully developed, just miniature version of a human that's inside a gamete and is just waiting to grow, literally get bigger. In this case, you can see that all of the genetic information would only come from one parent, so we already have an issue here. Although there were some caveats to explain that. Another example is blended inheritance. This is a case that's depicted by this little cartoon here where a spoon and a fork have a spork offspring. Even Charles Darwin believed a version of blended inheritance. He believed that there were little particles all throughout the body that came together in the gametes when they formed the offspring, such that all of these traits are mixed together and the offspring will have some sort of intermediate phenotype. And we now know that this is not true. Neither of these hypotheses turn out to be true. So what did we learn? What did Gregor Mendel's experiments tell us? So I just want to walk through a couple of experiments that Gregor Mendel did, and I'm going to say what he found, and then in parentheses here is now what we know, although he didn't necessarily make all of these leaps. He didn't know about DNA, for example, but what was happening? So just very quickly, what was he doing? He was crossing peas, right? So he had, for example, peas with a certain phenotype like green seeds and peas with a certain phenotype like yellow parents. And importantly, these peas and this parental generation or pea generation, you'll see me write later, are true breeding. That is to say that these peas, when selfed to themselves, which is a thing you can do with peas, will always form green. These peas, the yellow peas, when self to themselves, which I'm indicating with this little curved arrow, will always form yellow seeds. Now, in this first cross, you can see that when Mendel crossed the green and yellow parents, he got all yellow offspring. So here we can start to talk about some of the things that he found. So first off, you can see right here that these offspring do not take on an intermediate phenotypic value. Instead, there must be discrete units of inheritance. There must be some unit that confers this phenotype, or else they should have gotten some in-between color if it were a blended theory. We now know, Gregor Mendel did not know at the time, that these discrete units of inheritance are genes. So in this example here, we're looking at a case of the inheritance of different versions of this gene that confers the color in the pea seeds. And that brings us to our next point, is that there must be different versions of these genes. There's the green version and the yellow version, and we call these alleles. There's one gene that confers color, there's a green version, and there's a yellow version, or there's a green allele and a yellow allele. Another thing that Gregor Mendel was able to figure out is that the offspring must inherit one unit from each parent. That is, the parents have two copies and the offspring have two copies, and they got one from one parent and one from the other parent. What we now know is that this is true. Gametes are haploid. In a diploid organism, like me, in most of my cells, there are 2 copies of every chromosome, therefore 2 copies of every gene. But the gametes that I produce and that peas produce are haploid. These are the sex cells that are going to form offspring. Now, another thing that Gregor Mendel noticed, and you can see in the result of this cross here, is that sometimes one version of a gene will conceal or cancel out the presence of the other version, and this is what we know as dominance. In this case, there must be a green allele and a yellow allele. These offspring must have inherited both, that is, they must be heterozygous, having one of each version of the allele, but in this F1 generation, or filial, the first filial generation, it's what that F stands for, we only see the yellow phenotype. So what we can determine from this is that the yellow allele must be dominant over the green allele. Now, pay attention to what I'm saying there when I said that the yellow allele was dominant over the green allele. I did not say simply that yellow seed color was dominant to green seed color. Because again, dominance is this relationship between the alleles. The allele of one phenotype is what's canceling out the allele of the other phenotype. Now this last generation here, the F2 generation, which is the result of either selfing a P from the F1 generation or crossing basically sibling Ps in the F1 generation, is really important, and it helps solidify all of these points that Mendel learned. Really importantly, we see in the F2 generation that green comes back. This helps us know that there is a discrete unit of genetic information that confers this green phenotype, and it's not lost through these generations due to blending or anything like that. It can come back, and the ratio in which it comes back tells us about the underlying genetic mechanisms. So why peas? The first reason why peas were particularly well suited for this task is that you can either self-fertilize, because these individuals make both types of gametes, or you can cross different individuals by doing these pollen transfers. These peas that Mendel had access to also displayed many discrete traits and antagonistic pairs that he could analyze. So for example, there were differences in flower color, it was purple or white. Plant height, it was tall or short. Seed color, which we've been using, and a couple of others. This is really nice that these are discrete traits as opposed to continuous traits like, say, human height, right? That comes in on a continuous it continuously varies in the population. These traits were more one or the other, which made the types of analyses that Mendel was doing, looking at the ratios of these phenotypes, it made them a lot easier. It also, very importantly, means that these traits are very likely conferred by a single locus, or in other words, there is a single gene that confers this trait, and it comes in 2 alleles. That's not true with continuous traits. There could be many, many loci in the genome that contribute to that trait. Therefore, all of these ratios and these different crossing experiments would not be so straightforward. Also, you can quickly and easily grow a lot of peas. This is really important because the sample sizes that Mendel had to work with were really big in order to observe those ratios, to see that consistent 3-to-1 ratio that must have given him the aha moment. He had to observe a lot more than 4 peas, right? He had to observe hundreds and hundreds of them. So it was really important that his model organism, this pea, could grow quickly and he could achieve these great numbers. And lastly, for Mendel, he had easy access to these true breeding lines, or inbred lines. And this meant that there was this parental generation that he could tap into, where he knew that, say, the green peas were always going to produce offspring with green peas. So he could start with kind of a known genetic basis. He didn't know this at the time, but what he was working with were these homozygous genotypes, meaning that they had the same version of the gene in both copies of their gene. Now let's go back to that first cross and walk through it a little bit more specifically. So we saw that the parental generation that crossed between the green true breeding parents and the yellow true breeding parents resulted in all yellow offspring. One thing that we can take away from this is that the allele conferring the yellow phenotype must be dominant over the allele conferring the green phenotype. Or we can draw our first Punnett square, right? We know that the yellow parent, this true breeding parent, must be homozygous big Y, big Y. I'm saying a big Y to indicate this uppercase Y, and I've also color-coded it for ease of this particular example, but throughout this class you'll hear me say a big Y or little y to indicate the case, and that's me talking about which variant allele the phenotype is. So we know that this yellow true breeding parent must be homozygous for the allele that confers the yellow phenotype, that is, in our case, big Y, and I just made up this letter. We know that the true breeding green pea must be homozygous for the alternative allele, which I'm going to say is little y. Now, when we cross them, we fill in this Punnett square. What I'm putting on either the columns here or the rows is I'm separating out the two copies of this parent that the parent has into haploid cells. So for example, this homozygous big Y parent is going to produce half of its gametes that contain the big Y, and in this case, well, the other half are going to contain big Y too. But this will be not as straightforward, and we'll see in the next example. So what the Punnett square allows us to do is say what is the genotype of the offspring for every possible combination of parental gamete cells. So you can see in this case, they're all going to be heterozygous, right? Because these at this F1 generation can only inherit the big Y allele from one parent, and they can only inherit the little y allele from the other parent. Therefore, they will all be heterozygous. So we know that they're heterozygous, yet they display the phenotype of their yellow parent. This means that the big Y allele is dominant over the little y allele. Now, when Mendel crossed the F1 generation to each other, he saw that there were two different phenotypes, and this diagram already has them lined out as three separate peas. But remember that Mendel was looking at hundreds and hundreds of peas. He only saw the two phenotypes, but there was a very specific ratio that he cared about. He saw that there were 3 times as many yellow offspring as there were green. So what do we now know this means? Well, we can complete our Punnett square again, right? So now on the column and the row here, I am writing the gametes that can be produced by either parent. And since they are both heterozygous, they can either make big Y containing gametes or little y containing gametes. This is true for both of them. And now I start to see different things. When the big Y and big Y gametes come together, I get homozygous big Y, big Y. When the little y and big Y come together, I get the heterozygote. There are 2 ways to be heterozygous, depending on which parent you inherit which allele from. And then finally, you get the homozygous little y genotype if you inherit that allele from either parent. So this is where we get that 3-to-1 phenotype ratio, because from the last cross, we know that big Y is dominant. That means that big Y is going to be yellow, and the heterozygotes are going to be yellow as well, but only the homozygous little y is going to be green, resulting in a 3-to-1 ratio among offspring. I'm going to stop here, but I would like you to think about what would happen in a different combination of genotypes in the parents. Like say, for example, that one of these parents in this cross was the true breeding yellow. What would then the ratio of offspring genotypes and phenotypes be? And we'll pick up from there next time.